Hi, my name is Nabiha, and I'll be doing my presentation on directed evolution. Before we define directed evolution, let's first review what evolution is. The evolution of a species is the change in phenotype over time due to selective pressures. There are many different types of selective pressures, but the most common mechanisms of selection are natural selection and artificial selection. Natural selection is when individuals who are better suited for their environment are selected for. They're able to live longer and produce more offspring, thereby transferring their genetic material. Artificial selection is when humans choose for specific traits, either making them more common or obsolete. Humans using evolution to their advantage isn't a brand new or foreign concept. We actually use it all the time. A common example is selective pressure and breeding for the best crops. With all of this background information, we can finally tackle our original question of what directed evolution is. In the context of our course, directed evolution takes advantage of the principles of evolution to create macromolecules that can serve a specific purpose, or can be more efficient in their already ascribed purpose. So what is the purpose of this technique? As mentioned before, directed evolution is a method to create certain macromolecules that can accomplish human goals of creating certain products, catalyzing certain reactions, and learning more about genomes and evolution. This technique can be especially great because it takes advantage of microbes with small generation times. This saves us a lot of time, making this a very efficient process. Expanding on our purpose, there are many more specific goals we can accomplish using directed evolution. Directed evolution can be a method to create new drugs that can have a biological role in the human body. A method called phage display, which is a subset of directed evolution, is also used to make antibodies. There are also endless industrial applications for directed evolution experiments. Biomolecules perfected with directed evolution can make industrial processes much more efficient, environmentally friendly, cheaper, and selective. There have been studies on creating biofuel using directed evolution and ways to select for certain enantiomers in organic reactions with the method as well. Last but not least, directed evolution can be used to help us understand more about the processes of evolution and an organism's genome. Let's now look at how directed evolution experiments are conducted. As a general outline, we first begin by choosing our macromolecule, and then the process that we're interested in. The second step is to introduce mutations into the gene that encodes for this. Lastly, we allow the progeny to go through rounds of reproduction while screening and selecting for certain traits. Again, to begin this process, we need to choose a specific macromolecule. This macromolecule should be something that is close in function to the desired function. For example, let's say you wanted an enzyme that could break down alcohol, but you wanted it to work in an extremely low pH of 1. Alcohol dehydrogenase is an enzyme that has close to that function, but doesn't work so well in low pH. Perhaps this could be something we use directed evolution to create. If possible, before introducing mutations, you should really identify the genes involved in the function. If we know where the DNA encoding our function of interest is, we can be more mindful about where we put our mutations. Another thing to research is what key residues are integral in the function of the molecule. This can give us even more direction on what mutations will be useful. The next step is to actually introduce our mutations, or variations of the genotype that may affect our phenotype. Expanding upon points made in our last slide, if we know where our function is encoded, we would know where we want mutations in that certain area. Using a method called focused mutagenesis, we can do just that. There are multiple ways to go about doing this, but a common technique is to put degenerate codons, which can code for a wide array of amino acids, as the primary residues. Another method, if we do not know where to put our mutations that should ring a bell, is random mutagenesis. We can introduce random mutations to our genome using a variety of different methods, including chemical and physical mutagens that will affect DNA. We can also use faulty replication machinery to achieve this. Once we've achieved the mutations, we can allow our organisms with these mutated genes to propagate, and then screen and select for them as part of the evolutionary process. To review, screening is to take note of the different phenotypes in a population, while selection passes the different products through a filter to select one that fits our criteria. Both of these are used in direct evolution. Ways to screen include using fluorescent markers to mark activity, which can include the GFP we studied for much of the semester. Antibodies can also be used to mark protein levels, and flow cytometry can be used for intricate analysis of these cells. Methods to select for certain phenotypes include, but aren't limited to, affinity selection, compartmentalized self-replication, and compartmentalized partner replication. Affinity selection recognizes cells that bind to a specific target. CSR selects for DNA and RNA polymerase activity, while CPR selects more for Taq polymerase activity. This process is repeated over and over until our desired function is achieved, if it ever does get achieved. In the real world, this process has proven to be useful. A group of researchers at Caltech were looking for a new way to create carbon-silicon bonds, which are extremely expensive and costly to create. They ended up using directed evolution on cytochrome C from bacteria to create a metalloenzyme that is 15 times more efficient than industrial methods. This is a great example about how directed evolution can help us be more environmentally friendly and help us be more efficient in time, money, and energy. Thank you for watching my presentation. On this slide, you'll find all the references that I used to make this presentation. If you have any questions, please feel free to leave them in the discussion, and I will answer them to the best of my ability.